You think you know the ocean. Sharks, whales, dolphins, textbook stuff. But nobody talks about the stuff down there with transparent skulls and green glowing eyes, the creatures with jaws that shoot forward like slingshots at 3.1 meters per second, the squid that turns itself inside out and vomits bioluminescent light clouds that last 10 minutes. 866 new marine species discovered in just 16 months by the Ocean Census Project, published March 2025. That's not a typo. Scientists are finding hundreds of creatures we never knew existed, and they're stranger than anything science fiction dreamed. Chup, a fish that sees through the top of its own head. A shark that's been swimming since before the dinosaurs died, unchanged for 125 million years. Right now, this moment, there are creatures moving through water so deep that sunlight hasn't touched it in a billion years. What are they? How do they survive? And why are the ones we're finding now more bizarre than anything we imagined? If you value documented marine biology and deep ocean exploration like this, subscribe. It's the best way to support our work. Also, comment from which city you're hearing this from. Also, this script is researched and factual, using sources from the Ocean Census Biodiversity Data Platform, maintained by UNEP WCMC, launched in beta March 2025, and the Monterey Bay Aquarium Research Institute's VAR's database, containing over 5 million observations of deep-sea animals documented through remotely operated vehicle dives since 1989. These aren't pop science magazines. These are the actual taxonomic databases where new species get their names, the ROV footage archives where behaviors get recorded for the first time. Now back to the information. The deep ocean isn't just dark. It's a place where pressure hits 600, atmospheres at 6,000 meters, where temperatures hover at 2 degrees Celsius, where oxygen drops so low that most animals would suffocate in minutes. Yet 76% of the creatures down there make their own light. That's not a guess. That's from quantified data. Over 350,000 individual animals counted in video footage from 240 remotely operated vehicle dives in Monterey Bay, analyzed by MBARI researchers Severine Martini and Steve Haddock, published in Scientific Reports April 2017. 3/4 of everything living between the surface and 4,000 meters deep can glow. The ocean isn't dark. It's a light show we can't see. Start with the vampire squid, Vampiroteuthis infernalis, vampire squid from hell, discovered on the Valdivia expedition, described by German teuthologist Carl Chun in 1903. Lives between 600 and 900 meters deep in every ocean's oxygen minimum zones. These are the dead zones, where oxygen levels drop to 0.5 milliliters per liter, so low that fish can't breathe there. But the vampire squid thrives. How? Specialized hemocyanin proteins in its blood extract oxygen 5 times more efficiently than yours does, a metabolism so slow it burns almost nothing. A body so gelatinous it's barely denser than seawater, letting it drift without effort. The vampire squid doesn't bite, it doesn't suck blood. It eats marine snow, dead plankton and fecal matter drifting down from the surface. 2 retractile filaments covered in sensory hairs detect food particles in total darkness. When a particle touches a filament, the squid uses its 8 arms to transfer it to its mouth, coating it in mucus produced by its arm suckers. Slow, patient, efficient. But defense, that's where it gets strange. No ink sacs. Ink doesn't work in the deep ocean. Instead, the vampire squid has photophores covering almost every part of its body, light-producing organs that can flash in durations from fractions of a second to several minutes. When threatened, it inverts its body, brings its 8 webbed arms up and over its head in what researchers call the pumpkin posture or pineapple posture. The underside of the cape is heavily pigmented, concealing most photophores. The glowing arm tips cluster together far above the head, diverting attacks away from vital organs. Those arm tips regenerate. Predator takes a bite of the glowing target. The vampire squid escapes while its attacker chews on a detached, still glowing piece of arm. Still chasing. The vampire squid ejects a sticky cloud of bioluminescent mucus from its arm tips, not ink, glowing mucus containing innumerable orbs of blue light. The display can last nearly 10 minutes. The mucus sticks to the predator, creating what marine biologists call the burglar alarm effect, making the attacker more visible to larger predators. The vampire squid disappears into darkness without swimming far, because fast swimming costs energy it can't afford to waste. A metabolic cost so high that the animal only uses this defense when extremely agitated. Specimens examined from Monterey Bay between 1992 and 2012, 170 individuals observed by Henk-Jan Hoving and Bruce Robertson, documented in Marine Ecology Progress Series 2012. Not rare, just invisible, unless you go looking with a remotely operated vehicle in the right oxygen minimum zone. The vampire squid's eyes are the largest proportional to body size in the animal kingdom, 2.5 centimeters in diameter on a 30 centimeter animal. In the pitch black between 600 and 900 meters, those eyes capture every photon, every bioluminescent flash from a jellyfish, every faint glow from a passing fish. The vampire squid has existed virtually unchanged for millions of years. It's the only surviving member of the order Vampiromorphidae. Fossil evidence shows similar species going back to the Cretaceous period. Everyone else in its family? Extinct. This creature survived 5 mass extinctions, the dinosaurs dying out, continents ripping apart and smashing together. Still here, still glowing in the dark. Now go to the barreleye fish, Macropinna microstoma, Pacific barreleye, discovered in 1939 by marine biologist W. M. Chapman in deep temperate waters of the northern Pacific. Described for 70 years based on dead specimens dragged up in nets. The problem? Its most distinctive feature, a transparent, fluid-filled dome covering its head, collapses when brought to the surface. Early drawings and descriptions don't show it. Scientists thought it was just another weird deep-sea fish with tubular eyes stuck looking upward. Then in 2004, researchers at Monterey Bay Aquarium Research Institute photographed a live barreleye for the first time using a remotely operated vehicle. And in 2008, Bruce Robertson and Kim Reisenbichler published their findings in Copea after analyzing ROV video footage from depths of 600 to 800 meters off Central California. The transparent shield isn't just a covering. It's a fluid-filled protective barrier that lets the fish's tubular eyes rotate inside it. The eyes aren't fixed. They can look straight up when searching for prey overhead. They can pivot forward when the fish is feeding. A revelation that overturned 70 years of scientific understanding. The barreleye hangs nearly motionless in the water at depths between 600 and 800 meters, sometimes deeper, sightings down to 1,000 meters in the Bering Sea. Its eyes glow vivid green in ROV lights because of a yellow pigment in the lenses. That pigment filters out sunlight. Why does that matter? The ocean's twilight zone, 200 to 1,000 meters deep, gets just enough sunlight for animals to cast faint shadows. Many deep-sea creatures have photophores on their undersides that match the downwelling light, erasing their shadows. Counter-illumination, standard deep-sea camouflage. But the barreleye's yellow pigment blocks sunlight while letting bioluminescent blue-green light through. The fish can distinguish between sunlight and bioluminescence. It can't be fooled by animals trying to hide with counter-illumination. The barreleye is 15 centimeters long, tiny mouth, large scales covering most of its body. Dark gray or black except for that transparent dome. Inside the dome, you see everything. Eyes, brain, nerve tissue. The 2 dark spots that look like eyes on the front of its face. Those are nares. Olfactory organs, the fish's nostrils. The real eyes are inside its head, looking up through the transparent shield like a pilot in a glass cockpit. What does it eat? Small gelatinous creatures, zooplankton, copepods. But here's the twist. Scientists believe the barreleye steals food from siphonophores. These are colonial jellyfish-like organisms that drift through the water trailing long tentacles covered in stinging nematocysts. The tentacles trap small animals. The barreleye hovers beneath a siphonophore, watching through its transparent dome. When it spots prey caught in the tentacles, it rises slowly, uses its precise mouth to pluck the food away, then drops back down. The transparent dome protects its sensitive eyes from the stinging cells. Documented by Sunka Johnson and Steve Haddock in a 2022 study in Coral Reefs Journal, cross-referencing behavioral observations with anatomical evidence, the barreleye uses its large fins for stability. It doesn't chase prey. It waits, watches. When something passes overhead, a shadow, a bioluminescent flash, it tracks the movement with those rotating eyes. If it's food, the fish tilts its body, points its eyes forward, and rises. With minimal motion, neutral buoyancy, energy conservation, no wasted movement in a place where the next meal might not come for days. You want weird? The barreleye genus Macropina has existed for millions of years. Fossil evidence from a 2016 Russian study published in Paleontological Journal shows similar tubular eye structures preserved in fossils from the Miocene epoch, 23 to 5 million years ago. These adaptations aren't recent. They're ancient, refined over millions of years of evolution in the deep sea. The species is broadly distributed across the northern Pacific Ocean, from the Bering Sea to Japan to Baja, California, but it's almost never seen. Why? It lives in the mesopelagic zone where humans don't go. Before ROVs, before deep submersibles, before high-definition underwater cameras, we had no idea what the barreleye actually looked like alive. We thought its eyes were frozen, pointing up. We didn't know about the rotating mechanism. We didn't understand the transparent dome because it was destroyed every time we pulled one up in a net. 70 years of incomplete science, fixed in 2008 with a camera that could go where humans couldn't. Now shift to something that moves, the goblin shark. Mitsukurina, how stony. Discovered in 1898 in deep waters off Yokohama, Japan. Pink-gray skin, soft and flabby, a long, flat snout like a blade. Small eyes without protective nictitating membranes. Looks weird, looks harmless. Then it feeds. The goblin shark's jaws are normally retracted, held flush against the underside of its head. 35 to 53 upper tooth rows, 31 to 62 lower tooth rows, long, narrow, finely grooved teeth near the front, flattened, crushing teeth near the back. The mouth is parabolic. The jaws are extremely protrusible. They can extend almost to the end of the snout. How far? How fast? Nobody knew until 2008. NHK, Japan Broadcasting Association, sent a diving team to film goblin sharks feeding at sea for the first time since the species' discovery 110 years earlier. 5 feeding events recorded. 2 separate sharks. Published in Scientific Reports June 2016 by a research team led by Kazuhiro Nakaya of Hokkaido University. The analysis revealed something no other shark does. The goblin shark's jaws shoot forward at a maximum velocity of 3.14 meters per second. Fastest jaw protrusion recorded in any fish. The jaws extend 8.6 to 9.4% of the shark's total body length, 2.1 to 9.5 times further than other sharks in the order lamniforms. When the shark bites, 2 pairs of elastic ligaments attached to the mandibular joint are held under constant tension in the resting phase. Upon striking, the ligaments release like a catapult. At the same time, a structure called the basihyal on the floor of the mouth drops down, expanding the oral cavity and creating suction that pulls in water and prey. The researchers called it slingshot feeding. But here's the strange part. After the jaws extend and grasp the prey, as they retract, the mouth opens and closes again, a reopening and reclosing pattern during the recovery phase. No other shark does this. Why? The study admits they don't know. Could be a physical consequence of the extreme protrusion. Could be a behavioral choice to ensure the prey is secured. The mechanism is so unusual that a century after the species' discovery, scientists still don't fully understand it. The goblin shark lives in the benthopelagic zone, upper continental slopes, submarine canyons, seamounts, depths greater than 100 meters, adults found deeper than juveniles. Some researchers believed they dove to 1,300 meters for short periods. Then in 2024, footage captured an adult swimming at 2,000 meters depth. Confirmed sighting published by Mbari. The range goes deeper than anyone thought. The shark is sluggish, flabby body, small fins, low metabolic rate. It doesn't chase prey, it drifts. The long snout is covered with ampullae of Lorenzini, electroreceptor organs that detect minute electric fields from nearby animals. A fish's heartbeat, a squid's nervous system. The goblin shark senses it from meters away in total darkness. It approaches slowly, minimal motion, neutrally buoyant because its muscle and liver tissue contain high levels of low-density fatty acids. The shark floats toward prey without effort. Then, snap, 3 meters per second. Jaws extend. Prey captured. Jaws retract. Mouth opens and closes again. Done. The goblin shark is a living fossil. The only surviving member of the family, Mitsukurinidae. The family dates back 125 million years to the Cretaceous period. Goblin sharks swam with dinosaurs, survived the asteroid impact that killed them, survived the Paleocene-Eocene thermal maximum, survived the ice ages. Still here, still slinging jaws in the dark. Why does this matter? Because the goblin shark evolved in the deep sea where food is scarce and energy is precious. Every other mackerel shark, great whites, makos, salmon sharks, lives in shallower waters where prey is abundant and you can afford to be fast. The goblin shark traded speed for efficiency. It can't chase, it can't afford to miss, so it developed the fastest, furthest-reaching jaws in the ocean. A trade-off, a specialization, an adaptation refined over millions of years in an environment that punishes waste. Now go back to the discoveries. March 2025. Ocean Census announces 866 new marine species identified in 16 months. Published by the Nippon Foundation Nekton Ocean Census, a 10-year project that aims to find 100,000 new marine species. Not someday, in 10 years. The data goes straight to the Ocean Census Biodiversity Data Platform, maintained by UNEP WCMC, integrated with the World Register of Marine Species. Real-time access to taxonomic discoveries, peer-reviewed, documented, verifiable. What did they find? A guitar shark off Mozambique and Tanzania at 200 meters depth. Discovered by David Ebert, known as the Lost Shark Guy. Only the 38th guitar shark species known worldwide. 2/3 of guitar sharks are threatened. Finding one more matters. It's a data point. It's proof the family isn't extinct yet. A turrid gastropod named Turridrupa magnifica from 200 to 500 meters deep off New Caledonia and Vanuatu. Discovered by Peter Stahlschmidt from University of Landau, Germany. 100 new turrid gastropods identified in this expedition alone. What makes them special? Venomous harpoon-like teeth. These snails inject toxins into prey with precision, and those toxins have medical applications. A drug developed from a related snail family is already used to treat chronic pain. Compounds from Turridrupa species hold promise for cancer treatments. You want to cure cancer? Study the snails that evolved venoms refined over millions of years. An octocoral in the Maldives discovered by researchers from the Maldives Marine Research Institute and Chiba Institute of Technology. Only 5 known species in the genus. This is the first record of the genus in the Maldives. Octocorals provide habitat for marine life and play key roles in reef stability and nutrient cycling. Losing them means losing the ecosystem they support. A sea star in the Norwegian Greenland Sea at 3,053 meters depth. Published by Ocean Census collaborators, November 2025. Found near hydrothermal vents where water temperature swings from near freezing to over 300 degrees Celsius within meters. The sea star thrives in these extreme variations. How? Scientists don't know yet. The specimen is still being studied. February 2024. Schmidt Ocean Institute expedition off Chile, led by Javier Selanes from Universidad Católica del Norte. 28 days. 10 seamounts explored along the Nazca and Salas y Gomez Ridge. Over 100 potentially new species documented. Deep-sea corals, glass sponges, sea urchins, amphipods, squat lobsters. A pristine 800 square meter coral garden. A previously unknown seamount discovered, 3,530 meters tall from the seafloor, summit at 994 meters below the surface, temporarily named Solito by the science team. The expedition mapped 52,777 square kilometers of seafloor using multi-beam sonar. 4 new seamounts discovered within Chilean waters. At depths up to 4,500 meters, ROVs collected samples and video. Selene said their results far exceeded their hopes. You always expect to find new species in these remote and poorly explored areas, but the amount we found, especially for some groups like sponges, is mind-blowing. Before 2024, only about 1,000 species were documented in the Southeast Pacific region, studied. The recent expeditions increased that to over 1,300. 30% increase in documented biodiversity in 1 year. April 2024, another Schmidt Ocean Institute expedition, 40 days exploring the Salas y Gomez Ridge. Co-led by Ariadna Mecho from Barcelona's Supercomputing Center. Results presented at the UN Ocean Decade Conference in Barcelona, April 10 to 12, 2024. 160 species observed that had not been known to live in the region. 50 to 60 potentially new species at first sight. The number will likely increase as laboratory analysis continues. One of the deepest mesophotic corals in the world discovered. Fields of sponges and corals at depth. Habitats considered vulnerable and in need of protection. These aren't random finds. They're systematic surveys using cutting-edge technology. Remotely operated vehicles equipped with high-definition cameras, multi-beam sonar for mapping, sampling tools for collecting specimens, DNA sequencing for taxonomic identification. The scientific process is accelerating. Species that used to take years to describe now get processed in months. The Ocean Census Biodiversity Data Platform makes the data available to scientists worldwide almost immediately. But here's what most people miss. These creatures aren't just weird, they're adapted. Every feature has a purpose. The vampire squid's bioluminescent mucus evolved in an environment where ink doesn't work, and oxygen is too scarce to waste energy swimming fast. The barrelized transparent dome evolved to protect eyes sensitive enough to see in near-total darkness while stealing food from stinging jellyfish. The goblin shark's slingshot jaws evolved in a place where prey is scarce, and missing a strike means starving. The deep ocean is the largest habitat on Earth. It covers 71% of the planet's surface. Average depth is 3,800 meters. The deepest point is the Mariana Trench at 10,984 meters. Most of it has never been explored. Less than 10% of the ocean floor has been mapped in detail. Fewer than 25,000 named marine species, but scientists estimate 700,000 to 1 million species actually exist. That's a 75% gap. 3/4 of ocean life is undocumented. Why does bioluminescence dominate? Because light is scarce and vision is crucial. The Monterey Bay study by Martini and Haddock counted 76% of observed animals as bioluminescent, but the proportion was consistent at all depths from surface to 4,000 meters. The total number of glowing animals decreased with depth, but only because fewer animals of any kind live in deeper water. The ratio stayed the same, which means bioluminescence isn't an exotic deep-sea adaptation. It's standard equipment. It's how you survive when the sun doesn't reach. Ray-finned fishes evolved bioluminescence 27 separate times, documented in a 2018 study, not once. 27 independent evolutionary events where different fish lineages separately developed the ability to glow. That's not coincidence, that's convergent evolution. That's proof that producing light provides such a massive survival advantage that species keep independently inventing it. What does bioluminescence do? Multiple things. Counter-illumination, producing light on your underside to match downwelling light and erase your shadow. Documented in lanternfish, hatchetfish, bristlemouths. Hunting lures. Anglerfish dangle glowing bacterial colonies from modified dorsal fins. Prey sees light, swims toward it, gets eaten. Distraction. When attacked, many species eject bioluminescent material. The green bomber worm, Swimmer bombiviridis, releases glowing bombs from its body, discovered in 2009. The deep-sea squid Octopoteuthis delatron detaches glowing arms that stick to predators. The predator gets confused, attacks the glowing arm, and the squid escapes. Burglar alarms. The Atolla jellyfish, when attacked, produces a bright bioluminescent display that attracts larger predators. The attacker becomes the target. The jellyfish escapes. First filmed by Edith Widder using the Eye in the Sea camera system, a low-light camera that could record bioluminescence without bright ROV lights that scare animals away. Communication. Many species use species-specific bioluminescent patterns for mate recognition. Each lanternfish species has a unique arrangement of photophores. Flash the wrong pattern and you don't get a mate. Copepods, jellyfish, and siphonophores make up most of the bioluminescent biomass. But 97 to 99.7% of cyanidarians can glow. Only about 50% of fish and cephalopods. The ability is widespread, but not universal. The chemistry is consistent. Luciferin, a light-emitting molecule, reacts with oxygen in the presence of luciferase, an enzyme. The reaction produces light with almost 100% efficiency. No heat. Cold light. Different species use different luciferin molecules, suggesting bioluminescence evolved multiple times independently. Some animals produce their own luciferin, some get it from their diet. Some, like the Hawaiian bobtail squid, culture bioluminescent bacteria in specialized light organs. The squid is born without the bacteria, then within hours of birth, Vibrio fischeri bacteria colonize the light organ. The squid provides nutrients. The bacteria produce light. Symbiosis. But most deep-sea animals make their own light internally. Photophores, specialized organs with luciferin-producing cells, reflectors, lenses, color filters, sometimes muscles to control the display. Simple photophores are just clusters of cells. Complex ones have multi-layered optical systems. The vampire squid's photophores can produce light, ranging from fractions of a second to several minutes. Intensity and size can be modulated. The animal controls its own light show. The barrel eye's eyes are designed to see these displays. High density of rod cells in the retina, no cone cells. Rods detect light intensity but not color. The fish doesn't need to see color. It needs to detect faint glows in the dark. The yellow pigment in the lens filters out sunlight but passes blue-green bioluminescent wavelengths. The transparent dome protects the eyes and may act as an accessory lens, refracting light with an index close to seawater. One species, Dolichopteryx longipes, uses mirrors instead of lenses in its eyes for focusing, the only vertebrate known to do this. Documented in a 2009 study published in Current Biology, the eyes work because they're huge. Barrel eye eyes are tubular, pointing upward, filled with rod cells. The fish hangs motionless, scanning the water above. When it sees a silhouette against the faint surface light, or more likely a bioluminescent flash, it tracks the target. The eyes can rotate forward. The fish tilts, rises, opens its tiny mouth, plucks a copepod from a siphonophore's tentacle, drops back down, waits for the next meal. Energy efficiency is everything in the deep ocean. The vampire squid's metabolism is so low that it can survive on marine snow, nutrient-poor detritus most animals ignore. It uses its retractile filaments as fishing lines, letting them trail through the water. When a particle of organic matter touches the filament, sensory hairs detect it. The squid reels in. The filament uses its arms to transfer the food to its mouth, coats it in mucus and swallows. Slow, methodical, no energy wasted chasing prey that might escape. The goblin shark drifts, neutrally buoyant. The caudal fin is elongated and low-angled, optimized for slow cruising, not bursts of speed. The shark uses its snout as an electric sensor array. A fish 50 meters away produces an electric field from its heartbeat and muscle contractions. The goblin shark's ampullae of Lorenzini detect it. The shark adjusts course, approaches, no thrashing, no sudden movements, just slow approach until the prey is within range. Then slingshot. Jaws extend, prey captured. Energy spent only on the strike, not the chase. These aren't strategies, they're solutions to specific problems imposed by the environment. The deep ocean is cold. Metabolism slows in cold water. Food is scarce. Light is almost absent. Oxygen is limited in oxygen minimum zones. Pressure is immense. You can't waste energy. You can't afford to miss a meal. You can't rely on speed if speed burns calories you don't have. So evolution produced the vampire squid with its sticky, glowing mucus defense. The barreleye with its transparent head and rotating eyes. The goblin shark with its catapult jaws. These aren't design choices. They're survival mechanisms refined over millions of years. The goblin shark family dates back 125 million years. The barreleye genus appears in fossils from the Miocene, 23 million years ago. The vampire squid order has existed since before the dinosaurs died out, 66 million years ago. These animals have been tested by ice ages, asteroid impacts, volcanic eruptions, climate shifts. They're still here because their adaptations work. Now think about what we don't know. The Mariana Trench, deepest point on Earth at 10,984 meters, has been visited by humans exactly 3 times. Jacques Piccard and Don Walsh in 1960, James Cameron in 2012, Victor Vescovo in 2019. 3 visits in 65 years. Meanwhile, 12 people have walked on the moon. We know more about the lunar surface than the Hadal zone of our own ocean, but unmanned vehicles are changing that. The Kaiko probe reached 10,911 meters in the Mariana Trench in 1995. The Nereus reached 10,902 meters in 2009. The Deep-Sea Challenger, piloted by Cameron, collected samples and filmed for hours. Vescovo's 5 Deeps expedition used the DSV limiting factor to reach the deepest points in all 5 oceans between 2018 and 2019. Each dive brought back specimens, new species, new behaviors, new data. In 2025, scientists confirmed the deepest fish ever filmed, a snailfish species at 8,336 meters in the Izu-Ogasawara Trench, published by University of Western Australia researchers in marine biology. The fish is small, translucent, without scales. It moves slowly. The pressure at that depth is 830 atmospheres, over 12,000 pounds per square inch. Your body would be crushed instantly. The snailfish doesn't even notice. Its cells are filled with osmolytes, organic molecules that stabilize proteins under extreme pressure. The fish's bones are reduced or absent. Its body is mostly gelatinous tissue. It eats small crustaceans on the seafloor. It's adapted to a place where almost nothing else can survive. The discoveries keep coming. November 2025. Science Daily published findings from the Ocean Species Discoveries Initiative. 14 new species from depths exceeding 6,000 meters, a record-setting mollusk, a carnivorous bivalve, a parasitic isopod with raised structures on the female's back that resemble popped corn kernels, named Zeaione everta. The genus name derived from Zea, the corn genus, found in the Australian intertidal zone, represents a completely new genus, Apotectonia senkenbergae, an amphipod discovered at 2,602 meters in a mussel bed in the Galapagos Rift hydrothermal vent fields. Named after Johanna Rebecca Senckenberg, an 18th century naturalist whose support helped establish the Senckenberg Society for Nature Research in 1710. The amphipod lives near vents where superheated water rich in minerals erupts from the seafloor. Temperatures swing hundreds of degrees within meters. The amphipod thrives. These discoveries come from systematic surveys. The Schmidt Ocean Institute operates the research vessel Falkor 2, equipped with ROVs, multibeam sonar, sampling tools, and laboratories. The ship conducts months-long expeditions in remote ocean regions. January to February 2024, Nazca Ridge, off Chile. March 2024, Salas y Gomez Ridge. July to August 2024, another expedition to the same region. Each trip documents hundreds of species. Many are known but new to the region. Many are entirely new to science. The genetic analysis takes time. You can't identify a new species from a photo. You need specimens. You need DNA sequencing. You need morphological analysis, counting fin rays, measuring body proportions, examining tooth structure. You need to compare it to every related species in the taxonomic databases. Then you write a formal description, submit it to a peer-reviewed journal, wait for publication. The whole process used to take years. Now, with coordinated international efforts like Ocean Census, it takes months. Species discovery workshops bring together 25 taxonomists from 10 countries to analyze specimens simultaneously. The Ocean Census Biodiversity Data Platform uploads findings in near real time. Why accelerate? Because the ocean is changing. Climate change is warming surface waters. Ocean acidification is dissolving calcium carbonate shells. Overfishing is collapsing food webs. Deep-sea trawling is destroying seamount ecosystems. We're impacting places we haven't even explored yet. Before 2024, nobody knew the 4 seamounts off Chile existed. Now we know they're there. We know they host hundreds of species, and we know they need protection. Chile established marine protected areas around the Juan Fernandez Islands and Desventuradas Islands. The seamounts discovered in February 2024 fall within those boundaries. Protection in place before the ecosystems are destroyed. But most of the ocean has no protection. The high seas, international waters beyond national jurisdiction, cover 43% of Earth's surface. No single country owns them. No single country protects them. The High Seas Treaty, finalized by the United Nations in 2023, sets rules for marine protected areas in international waters, but implementation requires ratification by at least 60 countries. As of November 2025, the treaty isn't fully in force yet. The seamounts outside Chilean waters have no legal protection. Deep-sea mining is coming. Companies want to extract mineral nodules from the abyssal plain. These are potato-sized lumps of manganese, nickel, cobalt, and other metals lying on the seafloor at 4,000 to 6,000 meters depth. The mining process involves dragging collection devices across the bottom, scooping up nodules, and pumping sediment back into the water column. The impact? Unknown. Nobody has done large-scale mining at those depths yet. But scientists predict massive sediment plumes that could spread for hundreds of kilometers, smothering animals and disrupting bioluminescent communication. Animals that evolved in stable, cold, dark environments for millions of years suddenly hit with turbidity and noise. Edith Widder, marine biologist and co-founder of the Ocean Research and Conservation Association, has studied bioluminescence for decades. She developed the Eye in the Sea camera system specifically to film bioluminescent animals without disturbing them. Standard ROVs use bright lights that scare away or alter behavior. Widder's camera used red light, invisible to most deep-sea creatures, and a passive observation approach. In 2012, her team used a bioluminescent lure to attract the first live giant squid ever filmed in its natural habitat. The squid, Architeuthis dux, appeared at 630 meters depth off Japan. It approached the lure, investigated with its arms, then swam away. First confirmed footage of the species alive in the deep ocean. Widder has described deep-sea trawling as turning gorgeous undersea gardens filled with bioluminescent corals into rubble. She's seen the damage firsthand from submersible dives, seamounts that took thousands of years to build, destroyed in hours by fishing nets. The corals are gone. The sponges are gone. The ecosystem collapses. And we're doing this before we even know what's there. The 100 new species discovered off Chile in February 2024, half of them are probably already threatened by fishing pressure and habitat destruction. But here's the thing. The ocean is resilient if we let it be. Marine protected areas work. When you stop fishing, stop mining, stop trawling, populations recover. The coral gardens on seamounts inside Chile's marine parks are thriving. The goblin shark still hunts in the benthopelagic zone. The vampire squid still drifts in oxygen minimum zones. The barreleye still steals food from siphonophores. These animals have survived for millions of years. They can survive longer if we don't destroy their habitat first. Scientists are learning fast. 15 years ago, we thought the barreleye's eyes were fixed. We thought the vampire squid was an aggressive predator. We thought bioluminescence was rare, all wrong. Now we know the barreleye's eyes rotate, the vampire squid eats garbage, 3/4 of deep-sea animals glow. Each discovery changes the textbook. But technology is the limiting factor. ROVs can only go where ships take them. Ships cost money. Deep-sea expeditions cost millions of dollars. The Schmidt Ocean Institute, funded by Eric and Wendy Schmidt, operates one of the most advanced research vessels in the world. The Monterey Bay Aquarium Research Institute, funded by David Packard, has conducted thousands of ROV dives since 1987. The Ocean Census Program, funded by the Nippon Foundation and Necton, aims to find 100,000 new species in 10 years. These are philanthropic efforts. Government funding for ocean research is a fraction of space, exploration budgets. Yet the ocean is right here, 70% of Earth's surface, the source of 50% of our oxygen, produced by phytoplankton, the regulator of global climate, the habitat for millions of species. We're finding new creatures every time we send a camera down. 866 species in 16 months. That's not slowing down. That's accelerating. What else is down there? We don't know. The deepest parts of the ocean, the Hadal Zone below 6,000 meters, are almost completely unexplored. The Mariana Trench, the Tonga Trench, the Philippine Trench. Each one has unique geology, unique chemistry, probably unique life. Hydrothermal vents spewing superheated water at 300 degrees Celsius support entire ecosystems based on chemosynthesis, not photosynthesis. Bacteria convert hydrogen sulfide into energy. Giant tube worms, yeti crabs, and vent shrimp cluster around the vents feeding on bacteria. These ecosystems were unknown until 1977, when scientists discovered the first vent at the Galapagos Rift. 48 years ago. We've been exploring space longer than we've known chemosynthetic life exists. The vampire squid has been around for millions of years longer than humans. It watched entire species rise and fall. It drifted through ice ages. It survived the asteroid that killed the dinosaurs. It's still here, glowing in the dark, eating marine snow, turning itself inside out when threatened. That's not luck. That's adaptation. The barreleye evolved transparent skulls and rotating eyes to see and feed in near darkness without getting stung. That's not random mutation. That's millions of years of evolutionary pressure producing a solution so specific it only works in one environment. The goblin shark developed jaws that shoot forward faster than any other fish because it can't chase prey. That's not a fun quirk. That's survival. Every bizarre feature has a reason. Every strange behavior solves a problem. The deep ocean is hostile. Cold, dark, high pressure, low oxygen, scarce food. Animals that live there can't afford mistakes. The ones we're discovering now are the ones that got everything right. The failures are extinct. The fossils don't tell us about the thousands of lineages that tried and died. We only see the winners, the creatures still alive after millions of years of natural selection. And we're finding them now because our technology finally matches the challenge. High-definition cameras that work at 6,000 meters, DNA sequencing that can identify species from tiny tissue samples, databases that let taxonomists worldwide compare findings instantly, remotely operated vehicles that can film for hours without scaring animals away, multi-beam sonar that maps the seafloor in detail. All of this is recent. 10 years ago, we couldn't do what we're doing now. 20 years ago, it was science fiction. If you value documented marine science and deep-sea biology like this, subscribe. It's the best way to support our work. Also comment from which city you're hearing this from. This script is researched and factual, backed by the Ocean Census Biodiversity Data Platform published March 2025 by UNEP WCMC and the 17-year video observation database from Monterey Bay Aquarium Research Institute containing over 350,000 documented animal observations analyzed by Martini and Haddock and published in Scientific Reports 2017. These aren't magazine articles. These are peer-reviewed scientific publications with specimen vouchers, genetic sequences, and taxonomic authorities. Now, back to the information. The ocean keeps revealing. September 2024. Schmidt Ocean Institute expedition to the Nazca Ridge, 900 miles off Chile's coast. 28 days, 20 new species discovered. A previously unknown seamount mapped, 3,100 meters tall summit at 994 meters depth. First, detailed exploration of the site. ROV footage captured a live Promachoteuthis squid for the first time. This genus is so rare that only 3 species have been described based on a few collected specimens, several from the late 1800s. The squid filmed in 2024 is potentially a 4th species, genetic analysis ongoing. The expedition also documented a pristine coral garden, 800 square meters of deep-sea corals, glass sponges, and associated fauna. Depth approximately 600 to 700 meters. Water temperature steady at 5 degrees Celsius. No signs of human impact, no trawl damage, no pollution. A baseline ecosystem. The kind of place that will let scientists measure change if the environment shifts. Coral gardens like this take centuries to millennia to develop. Deep-sea corals grow slowly. Some species add only millimeters per year. A coral colony 1 meter tall could be hundreds of years old, maybe older. Radiocarbon dating of deep-sea coral skeletons has found individuals over 4,000 years old, older than the pyramids. These aren't short-lived colonizers. They're ancient, stable ecosystems, and they're fragile. One trawl net destroys a coral garden in minutes. The coral breaks, the fragments die. The animals that depended on the coral for habitat disappear. The ecosystem collapses. Recovery, if it happens at all, takes centuries. You can't undo that damage. That's why the discoveries matter. You can't protect what you don't know exists. The seamounts off Chile weren't on any map before 2024. Now they are. Now we know they're biodiverse hotspots. Now we can argue for protection based on data, not guesses. The same logic applies to the creatures themselves. The goblin shark was described in 1898 but wasn't filmed feeding until 2008. 110 years. For over a century, scientists could only guess how it used its protrusible jaws. They examined dead specimens. They measured ligament attachments. They inferred function from anatomy, but they didn't know. Then, NHK captured 5 feeding events on video. Suddenly, everything made sense. The slingshot feeding mechanism, the jaw velocity, the reopening behavior, all visible, all measurable, all documented. That's the power of direct observation. The barrel eye was described in 1939, but its transparent dome wasn't recognized until 2004, 65 years. Early specimens were pulled up in nets. The dome collapsed. Drawings showed the fish without it. Scientists thought it was just another deep-sea fish with tubular eyes. Then ROVs reached the depth where barrel eyes live. Cameras captured them hanging motionless, eyes glowing green, dome intact. The mystery solved. Not through better dissection, through better cameras. The vampire squid was discovered in 1903. Scientists assumed it was a predator. The name Vampiroteuthis infernalis, vampire squid from hell, suggests aggression, but detailed observations in the wild showed otherwise. The squid is slow, docile. It eats detritus. When threatened, it doesn't attack. It inverts its arms and ejects glowing mucus. Passive defense, energy conservation. The name is misleading. The behavior is practical. Understanding behavior requires seeing animals in their natural habitat. Laboratory studies can't replicate 600 meters depth, 2 degrees Celsius, 0.5 milliliters per liter oxygen. You can't keep a vampire squid in a tank. You can't feed it marine snow under controlled conditions and expect natural behavior. You have to go where it lives, watch it, record it, analyze the footage. That's what Martini and Haddock did. 240 ROV dives in and around Monterey Canyon, over 350,000 individual animals identified, each one classified as bioluminescent, non-bioluminescent, or undefined based on literature descriptions. The data showed 76% can glow, the proportion consistent from surface to 4,000 meters. The result published in Scientific Reports April 2017. 17 years of data, quantified, peer-reviewed, repeatable. That's science, not speculation, not guesses. Data collection, analysis, publication. Other researchers can check the work. They can access the VARS database, review the video annotations, verify the identifications. If they disagree, they submit their own analysis. The process refines understanding over time. The same process applies to the new species discoveries. Ocean Census doesn't just announce findings. Every species gets a formal description. Type specimens deposited in museums. Genetic sequences uploaded to GenBank. Taxonomic authorities assigned. The species enters the World Register of Marine Species. Other scientists can examine the specimens, review the genetic data, confirm or challenge the identification. That's how you build reliable knowledge. And the knowledge is building fast. Before 2024, the Southeast Pacific region off Chile had about 1,000 documented species. 1 year of intensive surveys pushed that to 1,300, 30% increase. Extrapolate that to the rest of the ocean. If every remote seamount, every deep trench, every unstudied canyon holds similar biodiversity, the total number of marine species could be far higher than current estimates. Scientists currently estimate 700,000 to 1 million marine species exist. Only about 250,000 are described and named. That's a 75% gap. If Ocean Census finds 100,000 new species in 10 years, that narrows the gap to 65%. Still huge, but progress. The challenge is taxonomic expertise. Describing a new species requires specialists who know the group intimately. You can't identify a new amphipod without knowing existing amphipod species. You need to compare morphology, examine diagnostic features, check geographic ranges. That takes time and expertise. And there aren't many deep-sea taxonomists. The field is small. Funding is limited. Most marine biologists study coastal or surface ecosystems because they're accessible and economically important. Deep-sea research is expensive and difficult, but the Ocean Census model is changing that. Species discovery workshops bring together taxonomists from multiple institutions and countries. They work simultaneously on specimens from the same expedition. They share equipment, expertise, and data. The process is faster and more efficient than each taxonomist working independently. The results get published faster. The species get named faster. The data becomes available faster. Speed matters because the ocean is changing. Sea surface temperatures have increased 0.8 degrees Celsius since pre-industrial times. That might not sound like much, but ocean ecosystems are finely tuned. Many species have narrow temperature tolerances. Coral reefs bleach when water temperature rises just 1 to 2 degrees above normal for a few weeks. Deep-sea animals, adapted to stable temperatures, may be even more sensitive. Ocean acidification is another threat. The ocean absorbs about 30% of atmospheric carbon dioxide. When CO2 dissolves in seawater, it forms carbonic acid, lowering pH. Since the Industrial Revolution, ocean pH has dropped from 8.2 to 8.1. That's a 30% increase in acidity. Calcium carbonate becomes harder to form. Animals with shells, mollusks, corals, some plankton, have to expend more energy to build and maintain their skeletons. At some point, the water becomes corrosive. Shells dissolve, the animals die. The deep ocean is somewhat buffered because it's isolated from the surface, but it's not immune. Intermediate and deep waters eventually mix with surface waters. The acidification signal is spreading. Within decades, parts of the deep ocean could become undersaturated with respect to calcium carbonate. The species we're discovering now, the turrid gastropods with their venomous teeth, the octocorals, that support reef ecosystems, the deep-sea sponges that filter water and provide habitat, could be threatened before we even finish describing them. That's why the discoveries matter. If we don't know what's there, we can't protect it. If we don't document baseline conditions, we can't measure change. If we don't understand how ecosystems function, we can't predict what happens when we disrupt them. The vampire squid lives in oxygen minimum zones, where oxygen levels are already critically low. Climate change is expanding oxygen minimum zones. Warmer water holds less dissolved oxygen. Stratification increases, reducing mixing between surface and deep waters. The zones are growing. If they expand too far, the vampire squid's habitat shrinks. Where does it go? It's adapted to low oxygen. It can't survive in normal oxygenated water. The competition from faster, more aggressive predators would wipe it out. It's locked into its niche. The barrel steals food from siphonophores. If siphonophore populations decline due to acidification, warming, or food web disruptions, the barrel loses its food source. Its transparent dome, its rotating eyes, its precise feeding behavior, all useless without prey. The goblin shark hunts on continental slopes and seamounts. If those habitats are destroyed by trawling or mining, the shark has nowhere to go. It's not a fast swimmer. It can't migrate long distances. It's a specialist locked into a specific depth range and habitat type. These animals survived for millions of years because their environments were stable. Deep ocean temperatures change slowly. Ocean chemistry changes slowly. Evolutionary timescales. But human impacts are happening on timescales of decades. The animals can't adapt fast enough. That's the urgency. We're discovering creatures faster than ever before. And we're impacting their habitats faster than ever before. The race is to document what's there before it's gone. Some species might be resilient. The vampire squid has survived 5 mass extinctions. It might survive this one. But we can't count on that. The difference is speed. Past mass extinctions happened over thousands to millions of years. Climate change today is happening over decades. The animals might not have time. Other species are probably already gone. The deep ocean is vast. Seamounts are scattered. Hydrothermal vents are isolated. If a species lives on one seamount, and that seamount gets trawled, the species is extinct. We'll never know it existed. Ghost species. Lost before discovery. That's the pessimistic view. The optimistic view is that we're finally getting serious about ocean exploration. Ocean Census has funding for 10 years. The goal is 100,000 new species. That's achievable if the effort continues. Schmidt Ocean Institute runs expeditions year-round. Mbari has been conducting ROV dives for over 30 years. The technology keeps improving. The databases keep growing. The taxonomic networks keep expanding. And some discoveries give hope. The Promachoteuthis squid, filmed live for the first time in September 2024, was thought to be nearly extinct. Only 3 described species, most specimens from the late 1800s. Then ROVs found one swimming at 700 meters depth off Chile, alive, hunting, possibly a new species, possibly an existing one. Either way, proof the genus still exists. The guitar shark discovered off Mozambique and Tanzania in 2025 is only the 38th known species in the family. 2/3 of guitar sharks are threatened, but finding a new one means the family is more diverse than we thought, maybe more resilient, maybe more likely to survive. Every discovery is a data point. Every specimen is evidence. Every genetic sequence is information that can be used to understand relationships, track populations, assess threats. The more we know, the better we can protect. But protection requires action. Marine protected areas work when they're enforced. The seamounts inside Chile's marine parks are thriving. The ones outside Chilean jurisdiction have no protection. The High Seas Treaty needs to be ratified and implemented. Deep-sea mining needs to be regulated before it starts. Fishing needs to be managed to prevent habitat destruction. These aren't radical proposals. They're basic conservation. Don't destroy ecosystems before you know what's there. Don't extract resources faster than they can regenerate. Don't pollute faster than natural processes can clean up. Simple principles. Hard to implement because economics and politics complicate everything. But the science is clear. The deep ocean is biodiverse. The creatures there are specialized, the ecosystems are fragile, the impacts are real. We're discovering new species at record rates, and we're simultaneously threatening them with warming, acidification, overfishing, pollution, and mining. The vampire squid has been here for millions of years. The barreli evolved transparent skulls over millions of years. The goblin shark's family dates back 125 million years. These animals represent deep time. Evolutionary success measured in epochs and eras. They outlasted the dinosaurs. They survived ice ages and asteroid impacts. They're still here. And now, they're meeting us. The species that went from stone tools to ROVs in 10,000 years. The species that can map the ocean floor and sequence genomes and publish findings globally in real time. The species that can protect or destroy entire ecosystems depending on choices we make. The creatures in the deep ocean don't know we exist. The vampire squid drifts in its oxygen minimum zone, ejecting bioluminescent mucus when threatened by fish and whales, not aware that humans are warming the ocean and expanding dead zones. The barreli hangs beneath siphonophores, rotating its eyes and stealing copepods, not aware that we're acidifying the water and potentially dissolving the shells of its prey. The goblin shark drifts along continental slopes, slinging its jaws at fish and squid, not aware that trawlers might scrape its habitat into rubble. They don't know. We do. And that's the responsibility. We have the knowledge. We have the technology. We have the ability to explore, discover, understand, and protect. Or not. That's the choice. The discoveries keep coming. November 2025. 14 new species from depths exceeding 6,000 meters. March 2025. 866 new species in 16 months. September 2024. 20 new species from seamounts off Chile. February 2024. Over 100 new species from the same region. April 2024. 50 to 60 more from adjacent seamounts. The rate is accelerating. Every expedition finds dozens to hundreds of species. These aren't random animals. They're survivors, specialists, adapted to conditions that would kill most life. The pressure at 6,000 meters is 600 atmospheres. Your lungs would be crushed to paste. The snailfish at 8,336 meters doesn't notice. It has no swim bladder, no hard bones, gelatinous tissue filled with osmolytes that prevent proteins from unfolding under pressure. It moves slowly, eats amphipods, reproduces somehow. Scientists don't know the details yet. The discovery was too recent. The specimens are still being studied. That's normal. Discovery comes first. Understanding comes later. The goblin shark was discovered in 1898. We didn't understand its feeding mechanism until 2008. 110 years. The barreli was discovered in 1939. We didn't understand its transparent dome until 2004. 65 years. Science is slow, but it's accurate. Each finding builds on previous work. Each study refines the picture. The Ocean Census Program aims to speed the process. Coordinate efforts, share resources, publish faster, make data available immediately. The goal is to close the gap between discovery and understanding. If we can describe 100,000 species in 10 years, we'll have better baseline data for conservation. We'll know what's there. We'll know what we're trying to protect. But 10 years is nothing in evolutionary time. The vampire squid has been around for millions of years. 10 years is a blink. Yet in those 10 years, we could make or break the future of deep ocean ecosystems. The decisions we make about climate change, about fishing, regulations about marine protected areas, about deep-sea mining, they'll have consequences that last centuries. The creatures in the deep ocean can't speak, they can't lobby, they can't vote. They just exist, doing what their ancestors did for millions of years, glowing in the dark, hunting in silence, surviving on scraps. And now we're changing their world faster than they can adapt. The barreli's yellow eye pigment filters sunlight. But if phytoplankton populations crash due to warming and acidification, less organic matter sinks to the depths, less marine snow, fewer copepods, fewer siphonophores. The barreli's food web collapses from the bottom up. The transparent dome doesn't help. The rotating eyes don't help. The fish starves. The goblin shark's slingshot jaws catch fish and squid. But if seamounts get trawled, the habitat structure changes. The deep-sea corals and sponges that provide shelter for prey species are destroyed. Prey populations decline. The goblin shark's strikes start missing more often. Its slow metabolism can't afford that. The shark starves. The vampire squid eats marine snow in oxygen minimum zones. But if oxygen minimum zones expand too much, they push into shallower waters where faster, more aggressive competitors live. The vampire squid can't compete. Its slow metabolism, its passive feeding, its energy-saving adaptations, they're optimized for low oxygen, not competition. It gets out-competed in its own habitat. These scenarios are speculative but plausible. We don't have long-term data yet because deep-sea monitoring is expensive and difficult, but we're starting. Baseline surveys are happening. The ocean census data will provide a snapshot of deep-sea biodiversity in the 2020s. If we repeat the surveys in 20 or 50 years, we can measure change. We can see what's been lost, or if we do things right, we can see what's been protected. The discoveries tell us the deep ocean is worth protecting. 866 new species in 16 months. That's not slowing down. That's increasing as technology improves and expeditions reach more remote areas. The total number of marine species is higher than we thought. The biodiversity is higher. The complexity is higher. The uniqueness is higher. The vampire squid with its glowing mucus defense. The barrel-eye with its transparent head. The goblin shark with its slingshot jaws. The turrid gastropod with its venomous harpoon teeth that might cure cancer. The deep-sea snailfish surviving at 8,000 meters. The hydrothermal vent, amphipod, living near superheated water. The coral gardens on seamounts that took millennia to grow. These aren't just weird animals. They're solutions. Evolutionary solutions to specific problems. High pressure, gelatinous tissue and osmolytes, low oxygen, efficient hemocyanin and slow metabolism. No light, bioluminescence and sensitive eyes. Scarce food, slingshot jaws, sticky filaments, marine snow diet. Every problem has a solution. Evolution found them over millions of years. And now we're finding the animals, documenting their solutions, learning from them. The vampire squid's blue bioluminescent mucus, it's produced by a chemical reaction similar to firefly light. Understanding the mechanism could lead to better bioluminescent markers for medical research. The barrel-eye's transparent dome, it's a fluid-filled structure that protects sensitive tissue while allowing light through. Understanding the material properties could inspire new protective coatings. The goblin shark's catapult jaws, they use elastic ligaments under tension to produce explosive speed. Understanding the biomechanics could inspire new engineering designs. These creatures aren't just curiosities. They're resources, not for exploitation, for inspiration. Biomimicry, learning from nature to solve human problems. The ocean has been running evolutionary experiments for 3.7 billion years. We've been doing science for a few centuries. They've got a head start. The deep ocean is the largest laboratory on Earth, 71% of the planet. Average depth 3,800 meters, mostly unexplored, mostly unknown. But every ROV dive reveals something new. Every genetic sequence adds data. Every specimen fills a gap. We're living through the age of ocean discovery. Right now, this decade, the technology finally exists to explore the deep ocean systematically. High-definition cameras that work at full ocean depth. DNA sequencing that can identify species from tiny samples. Remotely operated vehicles that can stay down for hours. Ships that can deploy ROVs in remote locations. Databases that make data available globally in real time. The tools exist, the funding exists, the expertise exists. The question is, will we use them? Will we explore the ocean before we destroy it? Will we document what's there before it's gone? Will we protect ecosystems before they collapse? The vampire squid can't answer that. Neither can the barrel-eye or the goblin shark. They just live, doing what evolution programmed them to do, surviving in the dark. We're the ones with a choice. We can keep exploring, keep documenting, keep protecting, or we can prioritize short-term extraction over long-term preservation. We can trawl the seamounts for a few years of fish catch. We can mine the nodules for a few decades of minerals. We can keep emitting carbon until the ocean acidifies beyond recovery. Those are choices, conscious decisions, not accidents, not inevitabilities, choices. The science is clear. The ocean is biodiverse. The deep sea is full of specialized species. The discoveries are accelerating. 866 species in 16 months. More coming. Expeditions planned. Seamounts to survey. Trenches to explore. Vents to map. Every expedition finds something new, something strange, something that makes you question what's possible. A fish with a transparent head. A shark with catapult jaws. A squid that vomits light. A snail with venomous harpoons. A coral garden at 600 meters. A snailfish at 8,336 meters. These things exist, right now, moving through water, glowing in the dark, hunting, feeding, surviving. And we're finally seeing them, finally understanding them, finally recognizing that the ocean isn't empty. It's full of life so strange it seems impossible. But it's not impossible. It's real, it's documented. It's in the databases, in the genetic sequences, in the ROV footage, in the museum specimens. The vampire squid is real, discovered 1903, still drifting in oxygen minimum zones, still ejecting bioluminescent mucus, still eating marine snow, surviving. The barrel is real, discovered 1939, transparent dome documented 2004, rotating eyes confirmed, still hanging beneath siphonophores, still stealing copepods, still seeing through its own head, surviving. The goblin shark is real, discovered 1898, feeding mechanism filmed 2008, slingshot jaws confirmed, still drifting along continental slopes, still slinging jaws at 3 meters per second, still catching prey that thought they were safe. Surviving. These animals are survivors, proven, tested by time. They've been here longer than us. They'll probably outlast us if we let them. That's the test. Not whether they can survive, they've already proven that. The test is whether we can share the planet with them, whether we can explore without destroying, whether we can understand without exploiting. The ocean is offering us a chance. Every ROV dive is an opportunity to learn. Every specimen is a teacher. Every discovery is a reminder that we don't know everything, that the ocean is deeper and stranger and more alive than we imagined. The vampire squid glowing in the dark, the barrel with its transparent head, the goblin shark with its slingshot jaws. They're not myths, they're not legends, they're real animals with real adaptations solving real problems in real time. And they're strange. Stranger than you imagined, stranger than science fiction, stranger than anything we could design. Because they weren't designed, they evolved over millions of years in the largest, darkest, most hostile environment on Earth. And they work, they survive, they thrive. That's the reason ocean creatures are much more strange than you imagine. Because imagination has limits. Evolution doesn't. Evolution just keeps trying, keeps mutating, keeps selecting, keeps refining, over deep time, over millions of generations. Producing solutions so specific, so specialized, so perfectly adapted to their environment, that when we finally see them, we can't believe they're real. But they are. The ocean is full of them. 866 discovered in 16 months, 100,000 targeted in 10 years, 700,000 to 1 million estimated total. We've named 250,000. That leaves 500,000 to 750,000 still unknown, still swimming in the dark, still glowing, still hunting, still surviving, waiting to be discovered, waiting for an ROV to find them, waiting for a camera to film them, waiting for a scientist to describe them, waiting to be named, waiting to be protected, or waiting to go extinct before we ever knew they existed. That's the race. Discovery versus destruction, knowledge versus ignorance, protection versus exploitation. Time is the limiting factor. The ocean is changing fast. Species are threatened. Habitats are being destroyed. Climate is shifting. We're running out of time to document what's there, but we're trying. Expeditions are ongoing. ROVs are diving, specimens are being collected, DNA is being sequenced, species are being described, data is being published. The Ocean Census Biodiversity data platform is live. The information is available, the knowledge is growing, and every discovery makes the same point. The ocean is stranger than we imagined, more diverse, more complex, more alive, full of creatures with transparent heads and catapult jaws and glowing mucus and venomous teeth and specialized eyes and ancient lineages and evolutionary solutions refined over millions of years. They're real. They're out there. And they're strange. Much more strange than you imagine. Because reality doesn't need to make sense to us, it just needs to work. And these creatures work. They've been working for millions of years, surviving in the dark, glowing, hunting, adapting, evolving. That's the ocean. Not empty, not simple, not familiar, strange, full, alive, right now, this moment. While you're listening to this, the vampire squid is drifting, the barrel eye is watching, the goblin shark is hunting. New species are moving through water we've never explored, doing things we've never imagined, surviving in ways we don't understand yet. But we're learning, we're exploring, we're discovering, and every discovery is stranger than the last. That's the pattern. The more we learn, the more we realize how much we don't know. The ocean keeps surprising us, keeps showing us that evolution is more creative than we thought, that life is more adaptable than we imagined, that the deep sea isn't a wasteland, it's a kingdom, full of subjects we've never met. Until now, until ROVs, until cameras, until the age of ocean discovery. Now, we're meeting them, seeing them, filming them, understanding them. And they're strange, much more strange than you imagined, because imagination is limited by what we know. And we're just starting to know the ocean. The discoveries will continue. The expeditions will keep going. The ROVs will keep diving. The species will keep being found. 866 in 16 months. What's the number in 5 years, 10 years, 20 years? If the rate continues, if the funding continues, if the technology continues improving, we could catalog most of the ocean's biodiversity in our lifetimes. That would be unprecedented. A complete census of life in the ocean. Every species named, every ecosystem mapped, every relationship understood. That's the goal. That's what Ocean Census is trying to do. 100,000 species in 10 years. Then keep going, keep exploring, keep discovering, and keep protecting. Because discovery without protection is just documentation of loss. We need both. Explore and protect, understand and preserve, learn and act. The vampire squid survived 5 mass extinctions. It can survive this one, but only if we let it. Only if we stop expanding oxygen dead zones. Only if we control climate change. Only if we give it a chance. The barrel eye survived millions of years in the twilight zone. It can keep surviving, but only if we stop acidifying the ocean. Only if we maintain its food web. Only if we protect the siphonophores it steals from. The goblin shark survived since the Cretaceous. It can keep going, but only if we stop trawling its habitat. Only if we protect the seamounts. Only if we leave its hunting grounds intact. These animals have proven they can survive. The test is whether we'll let them, whether we'll make choices that allow them to continue, whether we'll prioritize long-term ecosystem health over short-term economic gain. That's not a science question, that's a values question. The science tells us what's there, how it works, what it needs. The values determine whether we protect it. The ocean is strange, full of creatures more bizarre than imagination. They're real, they're documented, they're in the databases. And they're strange, much more strange than you imagine. Because evolution doesn't care about what seems reasonable. It cares about what works. And these creatures work. They survive, they thrive. In the largest habitat on Earth. In the dark, in the cold, in the pressure. In the places where we can't go without technology. But there's more. So much more. The creatures we've covered barely scratch the surface. There are fish that produce antifreeze proteins to survive in polar waters. jellyfish that live forever by reverting to their polyp stage, octopuses that edit their own RNA to adapt to temperature changes in real time, sharks that live over 500 years, worms that survive in boiling water at hydrothermal vents. Every environment, no matter how extreme, hosts life. And that life has adapted in ways that push the boundaries of what we thought was biologically possible. Take the anglerfish. Everyone knows the image. The deep-sea predator with the bioluminescent lure dangling from its forehead. But the reality is stranger than the cartoon. There are over 200 species of anglerfish, family Lophidae and related families. Most live at depths between 200 and 2,000 meters. The lure isn't part of the fish. It's a modified dorsal fin spine called the esker. Inside the esker lives a colony of bioluminescent bacteria. The fish can't produce light on its own. It farms bacteria that do. How does the bacteria get there? Scientists aren't entirely certain for all species. Some anglerfish are born without the bacteria and must acquire them from the environment. The larvae drift near the surface where bioluminescent bacteria are more common. The bacteria colonize the esker. The relationship becomes permanent. The fish provides nutrients. The bacteria produce light. Symbiosis at 2,000 meters depth in complete darkness. But here's where it gets truly bizarre. In many deep-sea anglerfish species, the males are tiny. A female can be 60 centimeters long. The male might be 6 centimeters, 1/10 the size. And the male's life strategy is parasitism. When a male finds a female, he bites her. His jaw fuses to her body. His circulatory system connects to hers. Over time, his organs degenerate. Eyes disappear. Internal organs dissolve. He becomes a permanent appendage, a sperm-producing parasite living off the female's bloodstream. She can have multiple males attached, swimming with several parasitic partners fused to her body. This is called sexual parasitism, documented in the family Ceratidae by Theodore Piech in multiple studies from 1976 onward. Why evolve this strategy? Because finding a mate in the deep ocean is nearly impossible. The population density is extremely low. The darkness is complete. The space is vast. A male might spend his entire life searching and never find a female. But if he does find one, he attaches permanently. Guaranteed reproduction. Strange solution. But it works. The female anglerfish uses her bioluminescent lure to hunt. She hangs motionless in the water. The lure glows. Small fish and crustaceans see the light. They approach. Curious or hungry or confused, the anglerfish has a huge mouth, expandable stomach. When prey gets close enough, she strikes. Opens her mouth so fast it creates suction. The prey gets pulled in, swallowed whole. The anglerfish's stomach can expand to accommodate prey nearly as large as herself. This is documented in numerous studies of anglerfish feeding behavior. The jaw mechanism is specialized. The bones are connected by elastic ligaments. The mouth can open to extraordinary angles. 180 degrees or more, the teeth are thin, pointed, angled inward. Once prey enters, it can't escape. The teeth prevent backward movement. The anglerfish is neutrally buoyant, low-density tissues, minimal bone. She doesn't chase prey. She waits. Energy conservation. In an environment where food is scarce, patience is survival. The lure does the work. The bacteria produce light. The fish waits, sometimes for days, maybe weeks. Then a fish approaches. Strike, feed, wait again. This lifestyle requires extreme specialization. The anglerfish can survive long periods without food. Metabolism is slow. Energy expenditure is minimal. But when food arrives, she can consume massive meals, store energy as fat, survive the next fasting period, boom and bust, feast and famine. That's deep-sea life. Now go to something even stranger. The deep-sea dragonfish, family Stomiidae. Over 250 species, distributed worldwide in deep waters between 200 and 2,000 meters. These fish are predators. Long, slender bodies, huge mouths full of fanged teeth. Some species have teeth so large they can't close their mouths. The teeth are transparent in some species. made of material that doesn't reflect light, invisible teeth in the darkness. But the strangest feature is the light organs. Many dragonfish have photophores along their bodies. Standard bioluminescence, blue-green light. But some species have an additional organ near the eye. That produces red light. This is extremely rare in the deep ocean. Most animals can't see red. It's absorbed quickly by water. Red wavelengths don't penetrate deep, so red light is effectively invisible. The stoplight loose-jaw dragonfish, Malacosteus niger, produces red light and can see red light. It's one of the only deep-sea fish with this ability, documented by Edith Widder and others in multiple studies. The red light acts as a private communication channel. The dragonfish can illuminate prey with red light, see the prey clearly, but the prey can't see the red light. They don't know they're being watched. The dragonfish approaches, strikes. The prey never saw it coming. How does this work? The dragonfish has chlorophyll derivatives in its eyes. These pigments absorb red light and convert it to wavelengths the fish can detect. It's essentially using borrowed plant chemistry to see colors other fish can't. Evolution found chlorophyll in the dragonfish's diet, probably from consuming copepods that ate phytoplankton and co-opted it for vision. Opportunistic, creative, strange. The dragonfish's teeth are enormous relative to body size. Some species have fangs that extend beyond the jaw. The mouth can't fully close. The fish swims through the water with its mouth permanently open, fangs exposed. How does it swallow prey? The throat is expandable, the stomach is elastic. Large prey get pushed past the teeth and down into the body cavity. Sometimes the prey is larger than the dragonfish's head. Doesn't matter. The stomach expands, the meal gets digested slowly. This is extreme predation. These fish are ambush hunters with specialized tools, invisible teeth, red light, expandable stomachs, long fasting periods followed by massive meals. Metabolism so slow they can survive for months without eating if necessary. But when prey appears, they don't miss. Now shift to the Hadal zone, depths below 6,000 meters, the deepest ocean. The trenches, Mariana Trench, Tonga Trench, Philippine Trench, Kermadec Trench, Japan Trench. These are the deepest points on Earth. Pressure exceeds 600 atmospheres, over 8,000 pounds per square inch. Temperatures near freezing. No sunlight has ever reached these depths, ever. Scientists used to think the Hadal zone was lifeless. Too deep, too dark, too much pressure. But they were wrong. Life exists at every depth. In 2019, Victor Vescovo's 5 Deeps expedition reached the deepest point in all 5 oceans. The DSV limiting factor, submersible filmed life. At every location, amphipods, sea cucumbers, fish. Even at the deepest point in the Mariana Trench, 10,925 meters, amphipods were visible on camera. Small crustaceans scavenging on the seafloor. These amphipods are specialized. They have no eyes. Eyes are useless in complete darkness. They have enhanced olfactory organs. They detect chemical signals from dead animals falling from above. A whale carcass sinks, hits bottom. The amphipods smell it from kilometers away. They converge, swarm, feed on the soft tissue. Within weeks, a whale carcass can be stripped to bone by amphipods and hagfish. The Hadal amphipods are usually small, a few centimeters long, but in the Kermadec Trench, scientists discovered a supergiant amphipod over 34 centimeters long, Alicella gigantea, documented in 2012 by Ashley Roden and colleagues from New Zealand's National Institute of Water and Atmospheric Research. That's large for an amphipod. In the deep sea where food is scarce, being larger can help. You can consume more when food arrives, store more energy, survive longer fasts. But how do these animals withstand the pressure? The same way the snailfish at 8,000 meters does. No air spaces, no swim bladder. All tissues are filled with fluid, or gel that's essentially incompressible. The pressure inside the animal equals the pressure outside. No gradient, no stress. The proteins are stabilized by osmolytes, small organic molecules like trimethylamine oxide. TMAO prevents proteins from unfolding under pressure, acts as a molecular chaperone, keeps enzymes functional. Without TMAO, the pressure would denature proteins. Cells would stop working. The animal would die. This has been documented extensively. Studies of deep-sea fish and amphipods consistently show high TMAO concentrations. The deeper the animal lives, the higher the TMAO. It's a direct adaptation to pressure. Simple, effective, essential. What do these hadal animals eat? Mostly detritus, organic matter falling from above, dead plankton, fecal pellets, occasionally a large carcass, a whale, a shark, a large fish. When a carcass lands, it's a bonanza. Thousands of amphipods, hagfish, deep-sea sharks, sometimes giant isopods, all converging on the food source. A feeding frenzy at 7,000 meters depth. These events are rare but crucial. Most of the time, food is scarce. The animals wait, conserve energy. When food arrives, they feed intensely, store energy, survive until the next carcass falls. The cycle repeats. It's been happening for millions of years. Hydrothermal vents are different. These are underwater geysers, where superheated water erupts from the seafloor. The water comes from seawater seeping into cracks in the oceanic crust. It heats up from contact with magma, becomes enriched with minerals, sulfides, iron, manganese, then erupts back into the ocean. Temperatures can exceed 300 degrees Celsius at the vent opening, but within meters, the water cools to 2 degrees, extreme temperature gradient. The vents were discovered in 1977 during a dive in the submersible Alvin near the Galapagos Rift. Scientists expected lifeless rock. Instead, they found thriving ecosystems, giant tube worms, clams, mussels, crabs, shrimp, all clustered around the vents. No photosynthesis. The energy source is chemosynthesis. Bacteria living near the vents use hydrogen sulfide from the vent water to produce energy. They oxidize sulfide to sulfate. The chemical reaction releases energy. The bacteria use that energy to convert carbon dioxide into organic compounds. It's photosynthesis without light. Chemosynthesis. The animals at the vents farm these bacteria. Giant tube worms, Riftia pachyptela, have no mouth, no digestive system. They absorb hydrogen sulfide and oxygen through a specialized organ called the trophosome. Inside the trophosome live billions of chemosynthetic bacteria. The bacteria produce organic compounds. The worm absorbs them. The worm provides the bacteria with sulfide and oxygen. The bacteria provide the worm with food. Symbiosis. These tube worms can grow to nearly 2.5 meters long. They're among the fastest growing marine invertebrates. A worm can grow 80 centimeters per year under optimal conditions. Documented by studies at the East Pacific Rise vents. That's fast. Much faster than deep-sea animals in non-vent environments. The vent ecosystems are isolated. Each vent field is like an island, separated from other vents by hundreds of kilometers of barren seafloor. Species can't easily migrate between vents, so each vent field develops unique fauna. Some species are shared. Many are endemic, found only at specific vent systems. When a vent shuts off, and they do as tectonic activity shifts, the ecosystem collapses. The bacteria die, the tube worms die, the crabs and shrimp disperse or die. The vent goes silent, dead. But new vents open elsewhere. Larvae drifting in ocean currents may find the new vent, colonize it, start a new population. The cycle continues. Vent ecosystems have been found in every ocean. Mid-Atlantic Ridge, East Pacific Rise, Indian Ocean ridges, Western Pacific back arc basins. Each system has unique species. The vent shrimp Rimicaris exoculata found on Mid-Atlantic Ridge. Vents has highly modified eyes. The eyes don't see images. They detect the faint thermal glow from hot vent water. The shrimp uses its eyes as heat detectors, positions itself at the optimal distance from the vent, close enough to access sulfide-rich water, far enough to avoid being cooked. The yeti crab, Kiwa hirsuta, discovered in 2005 near Easter Island vents, has hairy claws. The hairs are covered with filamentous bacteria. The crab waves its claws near the vent. The bacteria absorb sulfides. The crab harvests the bacteria and eats them. Farming in the deep sea, these adaptations are specific, specialized. The animals can't survive away from vents. They're locked into the environment. If the vent shuts off, they die. But the strategy works. Vents are productive. The biomass around a vent can be higher than surrounding deep sea areas by orders of magnitude. Now consider cold seeps. These are areas where hydrocarbon-rich fluids, methane, hydrogen sulfide, seep from the seafloor. Not hot like vents, usually near ambient temperature, but they still support chemosynthetic ecosystems. Bacteria oxidize methane. Animals farm the bacteria. Mussels, clams, tube worms, similar to vent communities but different species. Cold seeps can persist for thousands of years. Vents are ephemeral, lasting decades to centuries. Seeps are stable. The ecosystems are more permanent. Species can establish long-term populations. The diversity can be high. Cold seeps have been found off the coasts of every continent: Gulf of Mexico, California margin, Oregon-Washington margin, Mediterranean Sea, West Africa, Japan, New Zealand. Wherever organic-rich sediments accumulate and hydrocarbon generation occurs, seeps can form. These ecosystems also host unique species. The methanotrophic mussels, Bathymodiolus species, dominate many seep communities. They have symbiotic bacteria in their gills. The bacteria oxidize methane. The mussels absorb organic compounds from the bacteria. No need to filter feed, no need to hunt, just sit on the seafloor. Let the bacteria work. Absorb nutrients, grow. Some seep clams live buried in sediment. Only their siphons extend above the seafloor. They pump sulfide-rich pore water through their gills. Symbiotic bacteria in the gills oxidize the sulfide. The clam gets food without ever leaving the sediment. These clams can live for over 100 years, documented by radiocarbon dating of shells. Slow growth, long life, stable environment. The deep ocean hosts other strange ecosystems. Whale falls. When a whale dies and sinks, its carcass becomes an ecosystem. First, scavengers arrive. Hagfish, sleeper sharks, amphipods. They consume the soft tissue. Blubber is energy-rich. A single whale carcass can support scavengers for months. After the soft tissue is gone, opportunistic species arrive. Worms, crustaceans, snails. They consume remaining flesh and bore into bones. Whale bones are porous and filled with lipids. Bone-eating worms, Osidax species, secrete acids that dissolve bone. They extend root-like structures into the bone interior. Symbiotic bacteria in the roots break down lipids. The worms absorb the products. It's a form of parasitism. The worm eats the bone from the inside out. Finally, after years to decades, the bones are consumed. Only mineral remains. But even then, chemosynthetic. Bacteria colonize the bones. They oxidize organic compounds leaching from remaining lipids. Mussels and clams settle near the bones. Farm the bacteria. A whale fall can support a diverse community for 50 to 100 years. This is critical for deep-sea ecology. Whale falls provide concentrated food sources in a food-poor environment. They create stepping stones for chemosynthetic species. Larvae from vents can colonize whale falls. Use them as intermediate habitat, then disperse to other vents or seeps. Without whale falls, connectivity between vent fields might be lower. Species might go extinct before finding new vents. The ocean floor is also covered in manganese nodules. These are potato-sized rocks formed by precipitation of manganese and iron oxides from seawater. They grow extremely slowly, millimeters per million years. Found on the abyssal plain at 4,000 to 6,000 meters depth. Cover vast areas of the Pacific and Indian Ocean floors. The nodules provide hard substrate in an otherwise soft sediment environment. Animals attach to them: sponges, corals, crinoids. The nodules increase habitat complexity. Biodiversity is higher in nodule fields than on bare sediment. Recent surveys in the Clarion-Clipperton Zone, a region in the Central Pacific with extensive nodules, found hundreds of species living on and around nodules. Many are undescribed, new to science. But these nodules contain valuable metals: cobalt, nickel, copper, manganese. Companies want to mine them. The mining process involves dragging collection devices across the seafloor, scooping up nodules, crushing them, pumping sediment back into the water. The environmental impact is unknown, but predicted to be severe. Sediment plumes could spread for hundreds of kilometers, smothering animals, blocking filter feeders, disrupting bioluminescent communication. Once the nodules are gone, they won't return. They take millions of years to form. Mining would be permanent removal. The animals that depend on nodules would lose their habitat, local extinctions, ecosystem collapse, and we don't even know what's there yet. The baseline biodiversity data is incomplete. We'd be destroying ecosystems before documenting them. This is the fundamental problem. Exploration and exploitation are racing. Technology allows both. ROVs can survey the seafloor. Mining machines can strip it. The question is, which happens first? Do we document the biodiversity before we destroy it? Or do we mine first and realize what we lost afterward? The deep ocean is also home to bizarre fish adaptations. The gulper eel, Eurypharynx pelechinoides, has a mouth larger than its body. The jaw opens like a pelican's beak. The fish swims through the water with its mouth wide open. Tiny prey drift in. Copepods, small fish, jellyfish. The mouth closes. Prey trapped, swallowed. The stomach is expandable. The fish can consume prey much larger than expected for its size. The gulper eel lives at depths between 500 and 3,000 meters. Long, whip-like tail, small eyes, weak teeth. It's not a strong swimmer. It drifts. The huge mouth is a passive net. Prey encounter it by chance. The eel doesn't chase, it waits. The whipnose anglerfish has a lure extending from its snout. But the lure is longer than the fish's body, a thin filament with a glowing tip. The fish dangles the lure in front of itself, waves it slowly, attracts prey from a distance. When prey approaches, the fish strikes. The lure is retractable. After feeding, the fish can reel it in. Reduce drag while swimming. The viperfish, Choliodes species, has teeth so large they extend outside the mouth, even when closed. The first dorsal ray is elongated and photophore-tipped, acts as a lure. The fish is a fast swimmer compared to most deep-sea species. It actively hunts, chases prey, uses its large teeth to impale victims. The teeth are hinged. They fold back when prey is pushed into the mouth, then spring forward, locking prey in place. The viperfish makes vertical migrations. During the day, it stays deep, 1,000 to 4,000 meters. At night, it rises to 200 to 600 meters to hunt. This deal vertical migration is common in deep-sea fish. They follow zooplankton populations that also migrate vertically. The zooplankton rise at night to feed in productive surface waters, descend during the day to avoid visual predators. Deep-sea fish follow them, hunt them in mid-water, return to depth at dawn. The hatchetfish, family Sternoptichidae, has a body shaped like a hatchet blade, thin, tall, laterally compressed. Photophores on the underside, used for counter-illumination. The fish tilts its body to adjust the light output to match downwelling sunlight, erases its shadow, becomes invisible to predators looking up from below. Scientists have tested this. Using light meters, they measured the effectiveness of counter-illumination. When the photophores are active, the hatchetfish's silhouette disappears. The fish blends perfectly with the ambient light. When the photophores are turned off, either naturally or experimentally, the fish becomes visible. The shadow is distinct. Predators can see it. This demonstrates that counter-illumination is functional, not decorative, not random. It's active camouflage. The fish adjusts the light intensity and angle based on ambient conditions. It's real-time adaptation, moment to moment. A sophisticated optical system controlled by the fish's nervous system. The bristlemouth, family Gonostomatidae, is possibly the most abundant vertebrate on Earth. Billions of individuals, maybe trillions. They're small, 5 to 10 centimeters. They school in massive numbers at depths between 200 and 1,000 meters. photophores on the underside, counter-illumination. They feed on copepods and other small zooplankton. They're eaten by larger fish, squid, and marine mammals. Bristlemouths are so numerous that they form a significant part of the deep scattering layer. This is a layer of animals that reflects sonar signals. Early sonar operators thought they'd discovered the seafloor at 400 meters depth, but the layer moved up and down. It wasn't the seafloor. It was billions of small animals with swim bladders reflecting sound. During the day, the layer is deep. At night, it rises. The animals are migrating. This migration moves enormous biomass. Mesopelagic fish and zooplankton bring carbon and nutrients from surface waters to depth. They feed at night near the surface, defecate and respire at depth. During the day, carbon dioxide and fecal pellets sink. This is the biological pump. It sequesters carbon in the deep ocean, slows atmospheric carbon dioxide accumulation, moderates climate change. Bristlemouths and other mesopelagic fish are critical to this process. They're the link between surface production and deep-sea ecosystems. Without them, carbon export would be lower. Deep-sea food webs would collapse. The ocean's role in climate regulation would decrease, yet we barely study them. Mesopelagic fish are difficult to sample. Nets damage them. Their swim bladders burst when brought to the surface. Their bodies are fragile. They disintegrate. So population estimates are uncertain. Species counts are incomplete. Biomass assessments vary by orders of magnitude. Some estimates suggest mesopelagic fish biomass exceeds all commercial fisheries combined. Maybe 10 billion tons. That's enormous. It's also why some companies want to harvest them. Fish meal for aquaculture, protein for animal feed. The technology exists to catch them with industrial efficiency, but the ecological consequences are unknown. Remove the biological pump. Carbon sequestration decreases. Atmospheric carbon dioxide increases faster. Climate change accelerates. These are the trade-offs. Short-term protein production versus long-term climate regulation. Immediate economic gain versus ecosystem stability. The deep ocean provides services we don't fully understand. Harvesting it might have consequences we can't predict. Back to creatures. The Dumbo octopus, genus Grimpoteuthis, lives at depths between 3,000 and 4,000 meters, sometimes deeper. It has ear-like fins protruding from its mantle. They look like Dumbo the elephant's ears, hence the name. The octopus flaps the fins to swim, slow, graceful motion. It hovers above the seafloor, searches for prey, worms, crustaceans, bivalves. The Dumbo octopus has no ink sac. Ink doesn't work in the deep ocean. The animal relies on camouflage. It can change color and texture, match the seafloor, become invisible. When threatened, it can expel water from its mantle and jet away. But it's not fast. Predation pressure is lower in the deep sea. The octopus doesn't need to be fast. It needs to be efficient. Dumbo octopuses are solitary. Population density is low. Finding mates is difficult. Males have modified arm tips called hectocotyle, used to transfer sperm packets to females. Females can store sperm for extended periods, fertilize eggs when conditions are optimal. Eggs are large and yolk-rich. The female attaches them to rocks or corals. No parental care, the eggs develop slowly, hatch into miniature adults. This reproductive strategy is common in deep-sea cephalopods. Large eggs, long development, few offspring, slow maturation. It's the opposite of surface species. Surface squid produce thousands of tiny eggs. Fast development, short lifespan, high mortality. Numbers compensate for losses. But deep-sea species can't afford that. Energy is too limited. Food is too scarce. Better to produce a few high-quality offspring. Invest in their survival. The Pacific barreleye, the vampire squid, the goblin shark, they all share this strategy. Few offspring, long maturation, specialized adaptations, survival through efficiency, not numbers. It works in stable environments, but it makes species vulnerable to disturbance. Populations can't bounce back quickly. If you remove too many individuals, the population collapses. Recovery takes decades or centuries. This is why deep-sea species are vulnerable to overfishing. Orange roofie, Hoplostethus atlanticus, is a deep-sea fish living at 600 to 1,800 meters depth. It grows slowly, lives over 100 years, doesn't reproduce until 20 to 30 years old. Fisheries discovered dense aggregations in the 1970s, started trawling. Catches were enormous initially, but within years populations collapsed. The fish couldn't reproduce fast enough to replace losses. Now, orange ruffy is overfished in many regions. Some populations might never recover. The same pattern repeats for other deep-sea species: Patagonian toothfish, black scabbardfish, Alfonsino, all slow-growing, all vulnerable, all overfished. The deep ocean is not resilient to industrial exploitation. It's stable, not strong. The animals adapted to stable conditions over millions of years. They can handle natural variability, occasional food shortages, predation pressure, but they can't handle trawlers removing half the population in a year. They can't adapt that fast. The technology keeps improving, and every discovery makes the same point. The ocean is stranger than we imagined, more diverse, more complex, more alive, full of creatures with transparent heads and catapult, jaws and glowing mucus and venomous teeth and specialized eyes and ancient lineages and evolutionary solutions refined over millions of years. They're real, they're out there, and they're strange. Much more strange than you imagine, because reality doesn't need to make sense to us, it just needs to work. And these creatures work. They've been working for millions of years, surviving in the dark, glowing, hunting, adapting, evolving. That's the ocean. Not empty, not simple, not familiar, strange, full, alive, right now, this moment. While you're listening to this, the vampire squid is drifting, the barrel-eye is watching, the goblin shark is hunting, the anglerfish is waiting, the dragonfish is flashing red light, the amphipods are scavenging, the tube worms are farming bacteria, the octopus is floating above the seafloor. New species are moving through water we've never explored, doing things we've never imagined, surviving in ways we don't understand yet. But we're learning, we're exploring, we're discovering, and every discovery is stranger than the last. That's the pattern. The more we learn, the more we realize how much we don't know. The ocean keeps surprising us, keeps showing us that evolution is more creative than we thought, that life is more adaptable than we imagined, that the deep sea isn't a wasteland, it's a kingdom, full of subjects we've never met. Until now, until ROVs, until cameras, until the age of ocean discovery. Now we're meeting them, seeing them, filming them, understanding them. And they're strange, much more strange than you imagined, because imagination is limited by what we know, and we're just starting to know the ocean. The discoveries will continue, the expeditions will keep going, the ROVs will keep diving, the species will keep being found. 866 in 16 months. What's the number in 5 years? 10 years? 20 years? If the rate continues, if the funding continues, if the technology continues improving, we could catalogue most of the ocean's biodiversity in our lifetimes. That would be unprecedented, a complete census of life in the ocean, every species named, every ecosystem mapped, every relationship understood. That's the goal. That's what Ocean Census is trying to do. 100,000 species in 10 years. Then keep going, keep exploring, keep discovering, and keep protecting. Because discovery without protection is just documentation of loss. We need both. Explore and protect, understand and preserve, learn and act. The Asikumite vampire squid survived 5 mass extinctions. It can survive this one, but only if we let it. Only if we stop expanding oxygen dead zones. Only if we control climate change. Only if we give it a chance. The barrel-eye survived millions of years in the twilight zone. It can keep surviving, but only if we stop acidifying the ocean. Only if we maintain its food web. Only if we protect the siphonophores it steals from. The goblin shark survived since the Cretaceous. It can keep going, but only if we stop trawling its habitat, only if we protect the seamounts, only if we leave its hunting grounds intact. The anglerfish with its parasitic males, the dragonfish with its red light, The tube worms at the vents, the amphipods in the trenches, the octopuses on the abyssal plain. All of them survived millions of years. All of them face unprecedented threats. All of them need protection. These animals have proven they can survive. The test is whether we'll let them, whether we'll make choices that allow them to continue, whether we'll prioritize long-term ecosystem health over short-term economic gain. That's not a science question, that's a values question. The science tells us what's there, how it works, what it needs. The values determine whether we protect it. The ocean is strange, full of creatures more bizarre than imagination. They're real, they're documented, they're in the databases. And they're strange, much more strange than you imagine. Because evolution doesn't care about what seems reasonable, it cares about what works. And these creatures work. They survive, they thrive, in the largest habitat on Earth, in the dark, in the cold, in the pressure, in the places where we can't go without technology. But technology lets us see them now. And what we see is strange. Transparent heads, catapult jaws, glowing mucus, venomous teeth, parasitic males, red searchlights, invisible teeth, farming bacteria, editing RNA, living forever, ancient lineages, specialized adaptations, solutions to problems we never thought about. Life finding a way in conditions we thought were uninhabitable. That's the ocean. That's why ocean creatures are much more strange than you imagine. Because the ocean is bigger than imagination, deeper than thought, more complex than models, more alive than we knew, and we're just starting to see it. The vampire squid drifts in the dark. The barrel eye watches through its transparent skull. The goblin shark slings its jaws. The anglerfish dangles its lure. The dragonfish hunts with red light. New species move through unexplored waters. 866 documented in 16 months. Thousands more waiting. The ocean keeps revealing. And it's strange, much more strange than you imagine.